a black background with white writing that says Georgia and Kat's accessible audio-described science videos with an exclamation mark at the end of the sentence. The words Biology Core Practical 2 Enzyme Activity are under it.
Hello and welcome to our accessible audio-described science videos. I'm Kat.
And I'm Georgia.
Today we are going to be investigating how to measure the rate of enzyme activity.
There is a picture that is on the screen that is of all of the equipment that we are going to use for when we are going to do the experiment. There is a bottle that has got iodine in it, and there is a small beaker that we have already poured some of the iodine into, and there is a pipette that is next to it. There is also a spotting tile, which is a white square that has got wells that are dips, that are circular shapes inside of it. There are 12 wells. And then there is a bottle that has got the enzyme in it that we are going to use for the experiment, which is called amylase, and it is a clear liquid that is in another beaker that has got a pipette in it. We also have got some starch, which is a cloudy liquid that is in a beaker, and that has also got a pipette in it. The last things that we have got are a stirring rod, which is a long, thin cylinder that is made of clear glass, and we have got 4 boiling tubes that are empty and that are in a blue holder.
The first step is to put a drop of iodine into each of the wells of the spotting tile. This will turn blue-black if there is starch in the solution, and so we can use it to know if the amylase has broken down the starch, because the iodine won't change colour.
We are using a pipette to add the same concentration of iodine to each of the wells that are in the spotting tile. Some of the iodine, which is a solution that is an orange and brown colour, has gone on to the sides of some of the wells, but we are going to clean it away, so that doesn't matter.
Now that all the wells have got iodine inside of them, we are now going to add a set amount of starch and a set amount of amylase to 2 separate boiling tubes. It's important that they are in separate boiling tubes so that the amylase doesn't start to break down the starch before we're ready to start the experiment.
You can now see 1 boiling tube that is in the blue holder, and we are squeezing a pipette 4 times to add starch into it. And then we are using a different pipette 6 times to add the amylase into the other boiling tube.
We are going to get a stopwatch and mix the amylase and the starch in the same boiling tube. And after every 30 seconds, we are going to remove some of the mixture and add it to one of the wells to see if the amylase has broken the starch down.
On the screen, there is the spotting tile that has got the iodine inside of the wells, a stopwatch that is on a phone screen, and there are 2 boiling tubes. We are pouring the starch into the boiling tube that has got the amylase in it, and we are using a pipette to stir them, but we should have used the stirring rod to do that. The stopwatch is counting to the first 30 seconds, and when it has got to 30 seconds, we use the pipette to put some of the solution into one of the wells. We repeated that until we have added the solution to all of the wells, but the iodine goes blue-black because the amylase hasn't broken the starch down. Some of the starch and amylase solution has gone onto the table too.
Because the amylase didn't break down the starch in 6 minutes, or all the wells of iodine, we are now going to do the experiment again with a higher concentration of amylase.
We are doing the same things again that we had done when we had done the experiment for the first time. The iodine has stopped changing colour when we had been doing it for 1.5 minutes.
The iodine stops changing colour when the starch has completely broken down. In this experiment, we have shown that a higher concentration of the enzyme amylase breaks down the substrate starch much quicker. As well as testing enzyme concentration, you could also do this experiment by changing the pH by adding different buffers. You could also change the temperature by adding the enzyme and the substrate to a water bath at different temperatures. Both of these things would affect the rate of enzyme activity.
We hope that you have found this video accessible and interesting.
Please feel free to leave us a comment.
And don't forget to subscribe for more accessible content.